Okay, good afternoon once again, and welcome to your class in ATC 111, Crop and Animal Improvement. So last time, we just have our orientation. Good afternoon, lovely. Can you hear me?
Yes, ma'am. Good afternoon, ma'am.
Okay, so were you around last meeting?
No, ma'am.
Okay,
Ma'am,
just look for the under the course back. You just look for the orientation. We have recorded videos regarding how to use the Google Classroom, the requirements, and the course guide. So always look for the course guides here for the whole subject and for the whole semester. You can see everything here. So, and already I have posted your quiz, which is due today, but don't worry with time duration or deadlines, because you can always finish this throughout the semester, but meron lang siyang timeline or due dates just to give you um, reminders to do um, your phases or specific tasks to do. So good afternoon, Rich.
good afternoon, ma'am.
Okay, so this this call by the way is recorded. So I'm sorry for the last time because I missed the class and I had a field trip, as I have said. So we are now going through the module one, and some of you have already answered your quiz in advance, and I'm happy to know that, and your laboratory exercise as well. So this is the module one, and the quiz will cover everything here. So we'll start with the history of biotechnology. So along with crop and animal improvement, the latest technology that we have now is we call biotechnology. So in this presentation, um, you're going to you're going to know the different historical development defined by technology and genetic engineering process. Uh, and also what is the global status of the biotech crops. We call them biotech crops or genetically modified organisms, or what we call as genetic engineered crops or animals. So I bet that you know that since civilization began like 2000 years ago, so they say the earth or civilization have existed about 2000 years ago, and along with that is for hunting and gathering. So we call them pastoralism. So definitely they say that biotechnology is connected to years back when we do hunting and gathering. So humans as and early ancestors existed as food gatherers and hunters. So and when about 8,000 before Christ, the domestication of animals and the selection of plants began actually 8,000 BC or before Christ. So the Egyptians uh, master actually the art of wine making and baking leavened bread about 4,000 BC. So no una-una um kasi we'll say this is for they call this Mesopotamia or in Egypt for itself. We all know that the Middle East is the cradle of civilization. So doon kasi nag-start ang civilization. So, where it doesn't mean naman na doon nag-start ang civilization, wala pang tao sa Pilipinas. It's just that Well, maybe yes, wrong, or not. So, but one thing for sure is wala kasing records historically na meron ng mga what specific time meron ng tao sa Pilipinas, no? And of course, based on your knowledge, when does the Philippines discovered? Based sa knowledge ninyo, ha? Based on your knowledge, when does the Philippines discovered? By Magellan? Or was it 15 was it 1521? No, March 16, 1521. Amot lang ka. Ka-remember ba ko? Ka-remember mo? So, let's just proceed with, because maybe because of your connection, you cannot. Mary Rose, okay ka lang?
March 1521, ma'am.
Oh, yeah, so, do you really believe na 1520, iba pero pag 1521, naanay mga tao, naanatay mga deity, naanatay mga what do you call that, kanang communities, no? Meron na tayong mga communities, but prior to that, there must have been Filipinos already or early settlers before 1521. So anyway, to continue, on 1760s, it was marked as the age of industrial revolution. The use ng mga mechanical implements where the humans have able to use instead of hunting and gathering food, they now have the so-called machineries in agriculture and even crop milling. So that's 1760s. So of course, way far, wala pa tayong lahat niyan. And on 1860, there's already deliberate plant breeding and pollination of plants. No? So in 1860, meron ng, we have able to breed plants through pollination. And in the 1960s to 70s, it was marked as a period of agricultural advance known as the Green Revolution. So why Green Revolution? Because instead of existing na mga crops and animals, we use high-yielding varieties. We have able to choose na what are these good varieties and even use fertilizers and irrigation water and pesticides to increase crop yields and significantly strengthen food security. So, ayan. So, it's known as the Green Revolution wherein nakaila na sila o mga different breeds, hybrids, fertilizer, irrigation, pesticides, and so on. And also in 1953, because that's on the 1960s and 70s, as well as on 1953, if you know the following scientists, Watson, Crick, Wilkins, and Franklin, they elucidate the structure of DNA. So that was the time that they have able to identify the structure of DNA. And again, when we say DNA, deoxyribonucleic acid, it's the blueprint, the genetic makeup of our crabs, animals. So we humans also belongs to animals. So I just have to okay. So I will just check the attendance later, okay? But I don't know before 45 minutes, so mawala na pod mo. Anyway, to to continue, in 1983, meron na tayong first genetically engineered product called insulin. And of course, you know what insulin is, di ba? Para saan ang insulin, Jubert? Jubert, are you there? How about Reg?
Diabetes. Diabetes,
Control
ma'am.
production of sugar in the ano in the body. So in 1990, and as Reg said, it's used for diabetic person. And in 1990, meron tayong genetically engineered Bacillus thuringiensis cotton. So this BT cotton, which will be discussed in the next and under module 3 or 4, where we are going to discuss all the genetically modified organisms. So etong BT corn, meron siyang toxin from Bacillus thuringiensis, which kills the insect pest, no, nakakain or aatake sa cotton. So Kenneth, sorry Kenneth, kay naami group chat. Kenneth, are you there? Of course. So naami group chat and wala day ka sa group chat. So pero naa ka sa Google Classroom. So please ask your classmates what naa kay ko on Facebook, Kenneth. Okay, maybe he cannot hear me, so I'll just continue um and in 1990 the Human Genome Project was established. So this was a project to eventually map all the genes in a human.
Excuse me, ma'am.
Yes, Kenneth, na kay Facebook. Nama
Yes, ma'am.
o dili ta ka maa sa group chat, nami group chat. So pangitaan na lang ATC 111 na group chat. Pwede man nimo na i-search.
Okay, ma'am.
Okay, and then para ma-add ka dito or you ask na mag-add.
Okay, ma'am.
Ako ma-add imuhang, pangitaan ako, ang imuhang name lahi siguro,
Ken lang akong Facebook, ma'am.
letter as in letter N.
Ken. Ken lang.
I can. Okay. So I'll try later.
Sige lang, ma'am. Magpa-add lang ko sa mga classmate.
Sige, sige. So, okay lang ba sa inyo ang 2 p.m. nato nga meeting? Or would you rather have 1 p.m.?
Okay ng 2 p.m. ma'am.
Okay ra. So, kasi katulugon mangod ko medyo pag 1 p.m. In fact, katong last 5 nakatulog ko mo to na late ko gamay. So, but 2 p.m. okay na na pang pagising, no? But actually, as I have mentioned in our orientation, we will only have 45 minutes every meeting, and the rest are asynchronous. That means you're going to have to work on your laboratory exercise quizzes and advanced readings or late readings. No? So if you don't have more questions, then I continue with the topics that we have. 1994. Flavorsa tomato, I don't know if you have heard this one, is approved by the Food and Drugs Administration, making it the first biotech food crop na na-release sa market. So, if you're going to remember which is the first GMO na na-release sa market, so tomato yan, known as the Flavorsa TM, no, in 1994. And in 1996, biotech crops are already grown commercially on nearly 1.7 million hectares already, or 4 million acres worldwide. And this started actually in Argentina, Australia, Canada, China, Mexico, and United States mostly. Usually itong BT cotton. Now we have BT eggplant, which are already in trials in Philippines since like maybe 3 years ago. I remember that. And we have BT, we have been using already BT corn. No? So that's against corn borer. So in 1997, also the birth of the first cloned animal named Sheep Dolly was also gave birth to Dali. So much of the what do you call that, the descriptions and information about this GMOs will be discussed on the next chapters. So we'll be just talking about the timeline or development here. And in 2000, biotech crops are grown on already 44.2 million hectares or 100 million acres na in 13 countries. And first complete map of a plant genome known as the Arabidopsis thaliana. I don't know if you are familiar. Teresa, are you familiar of Arabidopsis? How about meron?
No, ma'am.
So, if you search through the Google, lahat ng mga researches are using Arabidopsis thaliana. Kasi ito yung unang tanim na first complete map na ng plant genome. Actually, by this time, human genome was already completely mapped along with different crops and animals. So we also have the so-called golden rice. This golden rice was started in the 1990s. This is believed to be available for developing countries to improve the health of undernourished people and also preventing blindness, no, for vitamin A deficiency. So ang white ginawang gold, more details of this will be discussed also. So in 2009, as shown on the graph here, biotech's graph are already present in 25 countries, 16 developing and 9 industrial countries. And it is actually the most rapidly adopted technology in the history of agriculture. So from 1996 to 2009 pa ang data, like live James, ayan, pa-increase siya ng pa-increase. So the scope of biotechnology involves the genetic engineering of animals, developing animal vaccines, so kasama na dyan ang COVID vaccine, and biocontrol agents against plants, pests, and diseases, plant protoplast fusion, so where a mutation occur, embryo transfer for animals, engineering ng plants to improve microorganisms. No, there are microorganisms na ginagamit natin for insulin production and even for cheese production at saka for sweetness, no, for example. And recombinant DNA for disease diagnosis, monoclonal antibody production, plant tissue culture, fermentation, no, ng wines, beers, cheese, and a lot of biofertilizers. So what is biotechnology if you're asked to? Of course, from the word bio, it means life, and technology means technique or procedure to develop new products. So it is the, in other words, the use of living organisms for their parts to make or modify products or improve plants and animals or microorganisms. So with this DNA, it can be modified to improve the crops and animals. So there are a lot of interdisciplinary areas, no, under this. So we have genetics, microbiome, biochemistry or chemistry, and even computer science, no, molecular bio, food technology and engineering, food science, biochemical engineering, breeding, and mechanical engineering. So recombinant DNA or genetic engineering is just one aspect of biotechnology, which are a set of technologies that artificially move functional gene species across boundaries to produce novel organisms. So in the genetic engineering, it is one of the most high technology of biotechnology. You will, in this procedure, the clone gene of interest is introduced into the cell, and the cell expresses the gene through the DNA transcription and RNA translation. So proteins are produced, and the trait is expressed. So what are the requirements for this? Of course, you need a vector or the one that contains the specific required characteristic, and the host cell usually is self-dividing and fast-dividing cells. And a way to introduce the gene into the cell, it's the procedure. There's a lot of procedure now, which will also be discussed in the next topics. And of course, you need time, a lot of time. No? To do this, traditional breeding would even take 20 years or more. But with genetic engineering, it also takes time, no? Because you need to have trials, a lot of trials, before it is released to the public. Although this could be like, if you notice, meron na tayong COVID vaccine in just less than a year already. No? So ganun lang kadali. But we can really say that it's already safe, no? But we should accept the facts since we really need in due time, so we just have to risk. So of course we need fundings, no? Big amount of money and a clone gene of interest. Kung ano ang kailangan, like for now, no? What do we need? So ano bang mauuna, yung higher yields, or we tend to go back, or we tend to choose genetic engineering to answer food security and malnutrition and poverty. So yun ang mga reasons natin. But we don't know yet the risk to come. Breeding with precision and accuracy of genetic manipulation. So when to use genetic engineering for the crop improvement, so these are the different uses na. If the trait of interest is not present in the crop germplasm. So if we want, like they have done in the BT corn, the one of the main problem ng corn is yung corn borer. So we have the specific bacteria, Bacillus thuringiensis, which are toxic to this pest. Now, once it is inserted to the genes of this corn, and the corn borer, as they attack the corn, they immediately are poisoned. So that's one of the trait of interest, and the trait is very difficult to improve using conventional method. So you use genetic engineering. So sometimes kasi we improve the breedings through traditional breeding methods na pollination breeding or manual or self-breeding. So improving the trait conventionally will also take a very long time, like 20 years, no, or forever. Genetic engineering allows the transfer of a specific gene of a desirable trait through the following. Now, this is just an example of how it was done. So again, you have the vector where you want the gene marker and the gene of interest. So you take the gene and then implant them into the bacteria. They used here agrobacterium, and through particle bombardment, and then infection is done through genes transferred to the plant tissue, and then it is selected and regenerated and then tested in the greenhouse and also in the field, and identify already the variety. Now this let's just say this papaya is usually virus, no? Virus resistant papaya. Parang PRSV papaya. The applications of biotechnology in the bioprocess technology, like the production ng essential primary metabolites, kagaya ng acetic acid, lactic acid, glycerol, and others. And the production of industrially used enzymes, lactases, production of monoclonal antibodies, vaccines, and novel recombinant products. So in the environment, meron din tayong different different na applications for waste treatment. We have a lot of plants that can remove toxics through waste bioremediation and also to control pollution, detect and monitor environmental contaminants, and recovery ng mga metals from mining waste and low-grade ores through phytoremediation accumulation. Sa plant biotechnology naman, we have a lot of genetically engineered plants for improved nutrition. We have, of course, again, the golden rice, which is nutritionally enriched in starch or amino acid and like the BT corn and the PRSD resistance ng papaya for insect pests and microbial diseases. And also stress tolerance, improved yield, drought-tolerant crops, aluminum and manganese tolerance for acidic soils, and improved post-harvest characteristic like the flowers of our tomato, and the genetic manipulation for cauliflower industry. So right now, we are very amazed with a lot of very beautiful breeds of colorful flowers already. Now, in plant biotechnology, again, another very practical, although high-tech din siya na application ng biotechnology ay through tissue culture. And we have used that extensively in banana and even orchids. And in biological control, like for BT insect control, rhizobium, nitrogen fixation, and bioherbicides. So in animal biotech, we also use genetic engineering for transgenic animal. Usually we use super mouse. It is the first GE animal with growth hormone genes. So madali lang siyang mag-grow. And introduce economically important traits into the commercial livestock, to improve quality of the meat, egg, wool quality, and quantity. So, mostly genetically engineered hormones and vaccines have been developed. And we have here the bovine growth hormone, somatropin or BST. It is the first biotech-derived pharmaceutical commercialized for animal agriculture, which are injected to cows for increased milk production. And of course, vaccines for animals are very common. So again, based nung sa na we have discussed, yung first commercially available na biotech crop ay flower saver, but for animals, it is the bovine growth hormone or the BST, the somatotropin. So sa healthcare naman, not only for animals but also for humans, we produce vaccines and monoclonal antibodies for hepatitis B vaccine. And for biopharmaceuticals, we have insulin for diabetics and somatostatin or growth hormone, interferons for cancer and virus diseases, and we call them gene therapy for the treatment of genetic disorders and disease cells. So more details of this will be discussed on the next meetings. High complex and potential could be realized in the near future also. So pagiging mas high-tech na ta karon, no? So this is actually a report on the global status of biotech, no, ni Dr. Clive James. As of 2009 pa niya siya. So as you can see, it's been increasing in the industrial and developing countries from 1996 to 2009. And in terms of global area, it's also been increasing. So pinakamataas ng increase ay soybean followed by corn and cotton and canola oil. And in terms naman ng pesticides, so herbicide tolerance is the highest, increasingly extensively, and insect resistance. But at the later of 2009, so yung herb tolerance and insect tolerance no, or resistance. So this is just to show you that mass between um, in case of as of 2010, in case of soybean, mas madami nang gumagamit ng biotech versus conventional. Sa cotton naman halos magkapare-pareho lang, but 49% na ay biotech. So sa maize naman, mas malaki pa din ang conventional but 26% are already using biotech. And sa canola also 21%. So by trait, mas gusto nila yung we have here BT and RR corn and Roundup ready ito. So ito yung herb resistant corn, yung herbicide resistant corn, and the BT corn or insect resistant. So as of 2008, so within any minute now, we have 10 minutes left. So after matapos, if maputol siya, then just go back to the link again to finally say the for questions and clarification lang. So, ayan. So, maraming, they usually BT corn yung mga ginagamit, but as of 2007 to 2008, marami nang gumagamit ng both BT and RR corn. So in the Philippines naman, so much of the adoption of the biotech corn is in Luzon, followed by Mindanao and Visayas. So in the Philippines, meron na tayong approved na for propagation na BTRR at saka BTRR corn. And also the use ng feed and processing corn, alfalfa, sugar beet, soybean, and potato, and even squash. So, kasi in the Philippines, I'll be presenting you in the end ng ating module kung paano ba papasok kasi although aside from usually ng mga biotech crops natin ay galing abroad, no? Although meron tayong sariling breeding system, but meron din tayong parang law on how to accommodate yung pumapasok ng mga GMO. So as for 2009, now we are 2020, so this has been increased extensively now. So 300,000 hectares already for highest BT and RR both. So 25, so yield ay tumaas ng 3.18 tons per hectare, which is 25% na biotech. So in the Philippines, we have the PRSV resistant papaya development, the BT eggplant, delayed ripening papaya, the golden rice, the BT cotton, and the virus-resistant sweet potato. Ito yung mga initiative ng Philippines contribution ng mga biotech crops. So this has been done by the Institute of Plant Breeding, the Philippine Rice Research Institute, kasi nasa atin yung IRI, International Rice Research Institute. So in fairness to the Philippines, kahit hindi tayo ang top producer ng rice, it is still Vietnam. But we do have, andito nag-host yung International Rice Research Institute. So which is now developing the golden rice. So Cotton Development Authority, or now PhilFIDA, and the Visayas State University. So if you have any questions regarding this topic, so you can ask questions, no? So next time, since we are running out of time, we're going to talk about the importance ng biotechnology. So, do you have any questions?
None so far, ma'am.
None, ma'am.
So, wala na yung mga pangutana. So, okay lang, next meeting na ta mag-discuss sa importance. So, one at a time na lang. No? Although, we're a bit late. No? Supposedly, by this time, we are done with the importance of biotechnology. But most of you, like Mary Rose, humana siyang answer sa Punnett Square. That's under genetics pa. So, hawod naman mo. So I hope you are already doing in your module 1 quiz, no? So these are the following questions. So may apat na naka-answer. No? And your exercise laboratory is the Punnett Square monohybrid and dihybrid cross. So if possible, meron kayong P1 parental 1 generation F1, F2, no, we include that in your monohybrid or dihybrid. Or monohybrid P1, first filial generation and second filial generation.
Excuse me, ma'am.
Yes. So,
Saan siya pag-demonstrate sa Punnett Square, ma'am?
actually, i-present ko pa yan dito sa Mendelian Genetics na na presentation. So, medyo advanced lang yung iba sa inyo, no? Kasi even you search that in the Google, makikita naman yan, no? Nakikita yan.
Gregor Mendel.
Yes, so ito. This is the monohybrid cross. So, pwede na ganun yung nikopyahon, no? But then, if you notice, ang double R round and yung small letter R are wrinkled. So, imuha lang na siyang ikuan sa box. So, i-pair lang mo big letter R and small letter R and so on. Makuha ba niyo, Kenneth, ang arrow? And then, R and R. Then R, big R, small R. So,
Yes, ma'am.
So in the P1 monohybrid cross, lahat sila ay apat na RR alike. They are genotypic all alike and phenotypic all alike. Kasi pareho sila ng result. Imuha na yan na i-pair-pair. So muna ang Punnett Square. Muna ginatawag siya Punnett Square. So iba din sa dito. Ito kanina, pareha ka sila big letter. Sa F1 monohybrid cross, kanina, capital R to and then small r. So, kung sa second or first filial generation ka na, gamit mo na yung RR. Ito na yung result ng parental, no, generation. So, ito na po din mo i-pair. So, first filial generation ka na, so, imuha ng i-pair ang duha. Since big letter R is brown and small letter R is wrinkled, and then, so kung R na siya, brown na siya, dili na siya wrinkled kasi recessive ang small letter. No? So less than a minute na lang. So i-pair na po na nimo. So big letter R and small letter R and big big letter R pero mauna gid ang first ang big letter, no? And then big letter R and small letter R and then small letter R and small letter R. So I hope you understand. Anytime na ta ha, maputol na ni siya. So do you have some more question?
Ma'am, okay pa dili mag-submit na ma'am, no?
Yeah, although tapos na ang deadline pero you have all the semester to do that. Pero
Okay, ma'am, thank you.
ayaw lang po paghulat og kuan ha kanang end of semester kay di na ka matulog ana. Pero although gilesson na ako actually ang mga requirements na po this time. So again, this is still under ano pa na chapter 3 and next meeting chapter 2 pa ta.
Ay hala ma'am. Karon ay humana din deadline na day ma'am. Kay ang nakabutang magud sa classroom kay karon adlaw.
Kaya pero laboratory kasi na siya, exercise. Okay lang na basag nila na siyang
Okay
deadline.
ma'am.
It's just a reminder na ay naday buhatonon. So wala man ay minus, no? Just like the attendance. Ang attendance ninyo gina-check lang nako para pang dagdag points. So if you notice yung grading system ninyo will be